Hello everyone, welcome to the cell biology series of the Biologics Everything Biological. So today we are going to study about lipid rafts and lipid droplets. So lipid rafts and lipid droplets are very important part of the plasma membrane and it is very important for CSIR net. So let's see what lipid rafts is. Despite their fluidity, lipid bilayers can form domains of different composition known as lipid rafts. It means the lipid rafts is a rigid structure in the cell membrane. The fluidity of the cell membrane is not same in throughout the membrane. Somewhere it gets rigid, somewhere it gets fluid. So how these rigid structures in the outer leaflet of the cell membrane form. So very important that these lipid rafts are mainly formed in the outer leaflet of the cell membrane, not in the inner leaflet. So due to the presence of the cholesterol and the sphingomyelin in the outer leaflet, they form a rigid structure. What happened here is that the bulk chain of the cholesterol and the long hydrocarbon chains of the sphingomyelin interact with each other and it forms a rigid and thick domain inside the cell membrane, the outer leaflet of the cell membrane, and it forms a lipid raft-like structure. So these lipid rafts are not only rigid, but because of its rigidity and the very high hydrophobicity, they can hold some of the major proteins and GPI anchors and many protein kinases which are very much responsible for the signaling mechanism. So you have to remember that the lipid rafts, what are present in the lipid rafts, mainly sphingomyelin and cholesterol. Other than this, glycolipids, as it is present in the outer leaflet, so glycolipids are also present, but majorly it holds the many proteins which act as a signaling relay center. Because of the high concentration of proteins present in the lipid rafts, they act as a signaling relay center for the signal transduction. So this is a very important part. From little bit of this information, you will get many questions that we will discuss in my later on lectures. Now, lipid droplets. Lipid droplets recent days is the hot cake of biological lipid research. So previously people are not that much worried about the lipid droplets, but these lipid droplets are mainly formed by the neutral lipids. Why these lipid droplets, how they form? So what we found that the membranes are mainly made up of lipids like sphingolipids, cholesterol and phospholipids. This among them, the cholesterols, sometimes the esters of these cholesterols are sometimes neutral, means they do not have any kind of hydrophilic heads like the normal cholesterol have, and the triacyl glycerols also does not contain any kind of hydrophobic heads. So what they did in ER, this cholesterol and this triacyl glycerols are processed, and what they does is that these neutral lipids get inserted inside this endoplasmic reticulum membrane, and these membranes finally form a circle-like structures, and they keep it and store it for further use during the other processes. Now you find that most of the cells stores an excess lipids in the form of lipid droplets because they want to retrieve it as a building blocks for membrane biosynthesis or as a food source. But other than this, these lipid droplets also play a major role in the development. So there are many OACs regarding the role of lipid droplets in the zebrafish embryo development, because these lipid droplets not only hold nutrients, but because of this hydrophobic nature, they contain some of the very important protein molecules which actually regulates the signaling mechanism. And you have to remember that these lipid droplets are packed in a single leaflet, not double leaflet. So what happened here, this cholesterol excess at endoplasmic reticulum forms and they contain in a single leaflet of phospholipids surrounded by single leaflet of phospholipids, and they get stored or they helps in the signal transduction. So you remember that these three-dimensional lipid droplets are actually covered by single stretch of phospholipids, and they contains neutral lipids inside these droplets like triacylglycerol and cholesterol esters, and they contain some of the protein, phospholipid monolayer protein and neutral lipids forms the lipid droplets. So you remember this, the function of lipid droplets and the role of lipid droplets in developments are very important, which some of the questions can come in the many entrance exams. So these are very important part. I have taken the help of the molecular biology of the cell through Salberts for the development of for the preparation of these slides, and the cell by Cooper. So if you like our video, please like, comment and share with your friends. Thank you so much, and please subscribe to our channel Biologics.